कौन है डॉक्टर प्रभा मैम? प्रभा मैम, Dr. Prabha ma'am, are you able to listen me? So ma'am, you will take something. Huh? Hello ma'am. Hello. Hello Dr. Prabha ma'am. Ma'am, so okay, okay. 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 So should I start class now? Okay, so let's start. Okay, let's start. Mm, Good evening, everyone. Hope you all are doing well and exciting. So let introduce myself. Myself, Kriti Ranjan, APMRF researcher scholar from IIT BHU. So I will take your this problem solving session for this course, computer aided drug design. So let's start now. Should I start? Please response. Okay. Okay. So let's come to the question number one. Drug metabolism predominantly takes place in. अरे यार ये होता रहेगा तो how I will take the class? What should I do? Should I wait sometimes to let people all in there? Huh? No, actually keep on people are entering now. So I have to allow them. So should I wait for them to enter or should I start my class? Ah, okay, let's start. You are able to see my slide? Okay, okay. Thank you. Okay, so let's start. Question number one. Drug metabolism predominantly takes place in. So, so first we will learn what is drug metabolism. So drug metabolism is what? Chemical modification of chemical modification of drug by biological enzymes to add its excretion by increasing its hydrophilicity. So why it's required? Because whenever we take the drugs or xenophytic compounds, so inside the body, uh, not all drugs or xenophytic compounds will reach to the liver. Sorry, to reach to the target site. So the rest one have to excrete from the body. So for that, the chemical modification occur. So it can easily can dissolve in water and get excrete out. So where it's occur in liver. Why it's occur in liver? Because there are lot of enzymes are available there which helps in chemical modification of drug. You people may know about the cytochrome P450. So okay, so let's see the options. First is kidney. Kidney basically excretion occurs. Pancreas and gastrointestinal tract, basically absorptions of drug occurs. You people must have heard about the ADME. ADME, like A for absorptions, D for distributions, M for metabolism, and E for excretions. So, whenever we take the drug orally, absorptions occur in a stomach, yeah, or we can say like gastrointestinal tract, and then from there, through blood, it reaches to the target sites, and from there, uh, some drugs will have to go for excretion. So for that metabolism, so in liver, that drug has to modify it so that it get excrete out. So answers will be here. So drug metabolism predominantly takes place in liver. Now come to the question number second. Tetracycline is used as, what is this tetracycline? This tetracycline is antibiotic. So it used as a antibacterial. So answer will be antibacterial. Now come to the question number three. A pharma company test 50 drugs. A pharma company tests 50 drugs on human volunteer. So probability of failure in phase 1 55%, phase 2 35.3%, and phase 3 45%. So how many drugs will successfully pass all the tests? So how we will calculate this? You all people are connected there? Okay, okay, okay. So how we will calculate this? So first we will find out so how many drugs has passed in phase 1. So for how we will calculate that? So how many drugs is there? 50 drugs. So we will take 50 minus 50 into 55 upon 100. So it will be how much? So it will be 22.5. So from 50, only 22.5 drug will reach to the phase 2, or this much drug has passed phase 1 stages. So this much drug only will will go for the phase 2 study. So in phase 2 study, again the probability of failure is 35.3%. So again we will calculate like how many drugs has passed phase 2 phase. So for again we will calculate similar like 22.5 minus 22.5 into 35.3 upon 100. So how much it will be? Yeah, it's good. 35.3 divided by 100. So 94. Yes. So it will be how much? 14 point. Sorry. So it will be 14.56. So from 22.5 drugs in phase 2, only 14.56 drugs has passed. So now we will check how many drugs has passed phase 3. So we will take this much drug. So we will take, sorry. So we will take this much drugs for phase 3 trial. So now this much 14. Sorry. So now we will take this 14.56 minus 14.56 into 45% has failed now. So we will calculate how much has passed in phase 3. So 45 upon 100. So it will come 8 drugs. So how many drugs will successfully pass all the tests? 8 drugs. Yes, ask. What you are telling? Like what? Yes. But in real life, how you will check the drug in real life? You will check the drug like this only in real life also. Now first you have to check drug in phase 1. So the drug which will pass the phase 1, that drug only will come to the phase 2. Yeah. I couldn't understand what you were trying to say. अरे लेकिन but like already WSL have is you this all process are independent. It's all are not connected, independent. Like the drug which will pass the phase 1, that only will come to the phase 2. It is not like that. Okay, phase 1, phase 2 will come first, then phase 1. Not like this. Okay. So let's come to the next question. Let's come to next question. Okay. Okay, so now question number 4. Failure of drugs during development is due to many reasons, such as poor biopharmaceutical properties, 40%, lack of efficacy, 30%, and toxicity, 45%. So, what is the percentage of drug that fail? So, how we will calculate this? The probability of failing assume as independent event. So, P failing equal to 1 minus P passing all the three. So 1 minus in due to poor biopharmaceutical property, it has failed 40%. So how much has passed? 60%. Here it has passed 70%, and here it has passed 55%. So we will calculate like this. So it will come 0.769. So percentage of failed drug would be 676.9%. uh, uh, can you uh, can you message me? Actually, I'm not able to hear. I will check there. but, but if these properties are not independent, we need to know about the intersection, don't we? But here in this question they are not asking about intersection. Yeah, yeah, but here I written now already like it's all at independent events. So how union will occur? Here intersection will occur, na? Huh? Huh, but these all are the independent events, na? Occurs due to many regions. So this can be the region, this can be the region, this can be the region, na? I couldn't understand. No, these all are the independent events. Don't know what you are trying to know. But it's yeah, but yeah, yeah. What you are telling is correct. But let us here we assume like these things are occurring as independent events. Okay, no problem. I will see this on next class. Next question. What is your problem? I will see that. Okay. Okay, so next, come to the next question. I have a transgenic mouse whose tumor is growing fast. So what type of drug can be tested with that mouse? Okay, so here option is anti-cancer, anti-diabetic, anti-inflammatory, and antipyretics. So, in which disease tumor used to grow? In cancer only. So, we will check first antipyretic drug. Antipyretic drug is the drug which used against the fever. So it won't be the options. Anti-inflammatory, it used against inflammation. So this also won't be the option. Anti-diabetic, it used against diabetes. So it also won't be the options. So answers will be anti-cancer because in cancers only the tumor formations occur. So if a transgenic mouse whose tumor is growing fast, so what type of drug we will give to mouse? Anti-cancer drug. So answers will be anti-cancer. Now come to the next questions. Which are the rotatable bonds? So let us first understand which all are the non-rotatable bonds. So unsaturated bonds, any single bonds connected to hydrogen or terminal atoms, single bonds of amides, sulfonamides, or those connecting two hindred aromatic rings are considered as non-rotatable. Okay, so now we will see in this structure. So let's see A. A is as terminal atoms. It is connected. It is a terminal atoms. So it won't be a rotatable bonds. And here this oxygen connected with this carbon by double bond. So this is also unsaturated bonds. So this also not be a rotatable bonds, and here also this is not a rotatable bonds, and this also not a rotatable bonds. So now we will come to C. See here this carbon connected with this oxygen by a single bond, so this will show the rotatable bonds, and here this carbon also connected with this oxygen by a single bond, so this also, so the rotatable bonds, and here also this carbon has connected with this carbon by a single bond. So this is showing rotatable bonds. So let's come to the option. Option A and C. A is I have told this is the terminal bonds. So it won't show the rotation, and so this is not with the option. Now come to the option C and D. Yes, in option C, this carbon is connected with oxygen by a single bond, and here this carbon also connected with oxygen by a single bond. So this both will show the rotation and will show the rotatable bonds, and here also this one will show the rotation or rotatable. This will be the rotatable bonds. So here option will be the C, D, and D. Okay, everyone. So now we come to the next question. How many rotatable bonds does these structures have? So as we studied in previous slide, these slides, like unsaturated bonds, any single bonds connected to hydrogen or terminal atoms, single bonds of amides, sulfonamides, or those connecting to hindered aromatic rings are considered as non-rotatable. So here we will see, like this carbon is like a terminal one, so it won't so rotate. It won't be a rotatable bond. This also won't be a rotatable bond. This also won't be. This also, and this also. So now we will see this all. So here we can see this carbon is connected with this NH bond by double bond, and this by single bond. So here resonance can be formed. So because of high energy barrier, here also rotation won't occur, and in this case also similar things occur. So here also because of this resonance. things, there also high energy barrier is there, so here also rotation won't occur, and similar case will occur here also. So in these three places also rotation won't be occur. So here only this carbon and nitrogen will show the rotation. So answer will be the A. Okay. So let's come to the question number 8. Cephalosporin C and amoxicillin. Cephalosporin C and amoxicillin mark which is not true. So, so these two are the antibiotics basically to the penicillin series. So let's see the structure of cephalosporin and amoxicillin. And this is the structure of cephalosporin and this is the structure of amoxicillin. And here first we will see what is the lactam ring. So lactam ring is a cyclic structure which basically contain nitrogen and ketonic groups, and as nitrogen connected to the beta atoms, uh, alpha beta positions, so it is called as beta lactam ring. So we will see in both antibiotics is there beta lactam ring or not. So yes, here in this cephalosporin, beta lactam ring is there, and in amoxicillin, here also beta lactam ring is there. So now we will see how this antibiotic acting on bacteria. So we will see first the structure of cell wall structure of bacteria. This is basically made up of peptidoglycan. This peptidoglycan is made up of NAM, and NAM, what is NAM? NAM is like N-acetyl muramic acids, and NAM is N-acetyl glucosidic muramic acid and glucosidic acids. Glucosamine, sorry, glucosamine. So this is basically glucosamine and N-acetyl glucosamine, and another one is N-acetyl muramic acid. So this N-acetyl glucosamine connected with another N-acetyl glucosamine by a transpeptide bond. So what will happen? This antibiotic will come and attach to the active site of this transpeptidase and inhibit its activity. So once it will inhibit this activity, so the connection between them will break or it won't form. So cell wall synthesis will stop. So now come to the option. Cephalosporin C and amoxicillin mark which is not true. So yes, this is antibiotic, can treat bacterial infections because once it will bind, it will not allow cell wall synthesis. So of course, it will stop the bacterial infection. So this option also be a correct. And now the option number C do not have the beta lactam ring. Yes, it's having beta lactam ring as I have showed in both antibiotics. So this is the mark which is not true. So this one is the answer. Yes, prevent the cell wall synthesis. Yes. So answer will be the C. So now come to the question number 9. In this list, which drug is given as IV? IV means intravenously, as in injection form. So let's come to the options. A. Ibuprofen. B. Atorvastatin. C. Aripiprazole. And D. Rituxan. So ibuprofen, we know this is the painkiller and used as a pills. And this atorvastatin, this is a cholesterol degrading drug, and it also used as a pills. Aripiprazole, it also used as a as pills and used against the schizophrenia. And this is the only drug which is an anti-cancer drug and used in an injections form or with, giving as intravenously. So answers will be Ritujan. So now the question number 10. Any question? So now this is the question number 10. Why should drug be soluble at pH 2 to match? Option A, pH of blood. Option B, pH of intestine. Option C, pH of stomach. And and option D, pH of kidney. So let's know the pH of all these. So pH of blood is how much? Yes, good. Yes, the pH of blood is 7.4. pH of intestine is also 6.4 to 6.4 to 7. pH of a stomach is 1.5 to 1.8 to 3 nearly, and pH of kidney is also 7.3 to 7.4. So why should drug be soluble at pH 2? So you all know, like when we take drug orally, it will come to the stomach. So drugs, as we know now, the pH of the stomach is 1.8 to 3. It is the acidic pH. So if the drug is soluble at pH 2, then only it will solubilize here, or it will be stable here in the stomach. So that's why the drug should be soluble at pH 2. So answer will be the C, pH of stomach. Let's do some more questions. This all, I think, your assignment questions. So how many rotatable bond does these structures have? Tell. Sorry, write in there. Uh, this can use na Ritujan. This is anti-cancer drug na. Just now I told you. So now question number 5. Where this in this question. No. In my question, it's not like tumor is growing parts of what type of drug can be tested. No, so anti-cancer drug. Just now I told no. Hey, I don't know about your assignment. What can you write it down here because I don't know. Yeah, tell me, ma'am, again. that's why I told you one example is there na in this option itself. This one, Ritujan. This is the drug. Yeah, yeah, a lot of drug is there. Right now, I don't remember many, but I can tell. Yeah, cisplatin. Most of like the cisplatin, famous one is there. Cisplatin. You people, you may know. Okay, okay, Ranjan Sahu. I will repeat first. Should I completed this presentation? So the question number 11: how many rotatable bond these structures having? So I told previously, the terminal atoms won't be. So the rotatable bond. So this won't be. This won't be, and this, and this, and even this. So how many rotatable bonds is here? This carbon and this carbon one, this two, and then this carbon connected with this oxygen three, this carbon connected with this oxygen four, and this carbon with this five, six, seven, and eight. So answer will be the eight. So here eight rotatable bonds in this structure. So now the question 12. How many rotatable bonds does these structures have? So in this structure also, this is a rigid bond, so it won't rotate. And this is the terminal bonds, so it also won't show the rotation bond. This also and this also. So here also this carbon connected with this oxygen will show the rotation and show the rotatable bonds. This is the rotatable bonds. This is the rotatable bonds. This is the rotatable bonds and this is the rotatable bonds. So one, two, three, four, five, six. So answer will be the six. So what is? Hey, let's do you people na this question. Let's do what is meant by lead compound in medicinal chemistry. Options A: drug containing the element lead, a leading drug in a particular area of medicines, a compound that acts as the starting point for the drug design and development, or a drug which is normally the first to be prescribed for a particular element. So tell what? Yes, yes. Oh, correct. Good. Or you can, okay, correct. Compound that act as the starting point for the drug design and development. Now this question 14. Which of the following need to be established before the search for a lead compound takes place? Option A, the pharmacophore. Options B, a structure activity relationship. Option C, a bioassay. And option D, patents. So what would be the answer? So, so which of the following need to be established before the search of? No, no. So before, yeah, structure activity relationship, then only you will search na lead compounds, because first you have to check na how these compounds are binding. So a structure activity relationship. What is the term used for the automated in vitro testing of large number of compounds using genetic? Hey, what happened? So what is the term used for the automated in vitro testing of large number of compound using genetically modified cells? What is the term used for the automated in vitro testing of large number of compounds using genetically modified cells? So option A, robotic testing, option B, high throughput screening, option C, multi screening, and option D, nanotechnology. So what would be the answer? Yes, high throughput screening. Okay, so for today, this much only I have made, I think. Thank you. They have asked no something. Failure of drug questions. Which one? This one, Ranjan? No, so this very clear now. This one. So what doubt you have in this? 76.9. And here we have to consider these all properties as independent event. Then only we can calculate how much percentage of drug that failed. Huh? This one. Okay, so this much came 0.769. Arey, write it down, message, write it down. Then only I will understand. Okay, write it down. Question number 13 once. What is question number 13? What is meant by a lead compound? Lead means, uh, okay. Lead compounds we need na in medicinal chemistry. Lead means a compound that will not heat compound. In heat, like we are taking large number of compounds and the compound which showing maximum activity against the target that will take as a lead compound. So what would be the lead? Then only we can start drug design and development. Na? So just any compound that act as the starting point for drug design and development, that would be the lead compound in medicinal chemistry. These all are the independent properties only, na? Do you think that drug which passed phase 1 are not more likely to pass phase? No, no, that drug will pass. कहाँ गया? Some will. हाँ. उसको. बोल रहा है. Just a while ago that drug which passed phase 1 are more likely to pass phase 2 than those which did not. This make events in passing phase 1. The end of seventh question mentioned that amide groups as non-rotatable bonds, and seventh question saw amide bond with carbon. Then how there is? I couldn't understand what they're asking now. हाँ tell. Non-rotatable bonds. एमाइड कहाँ है? Met metformin. I think in this Sanat Kumar metformin. I think, or let me check once. I don't remember. Oh, sorry, not metformin. Yeah. It's correct. Not metformin. Yeah, this is correct one. Yeah, correct. You people correct. Yes. This one is the only paclitaxel. Yeah. This metformin is anti-diabetic drug. I got confused. Question number third. Yes, tell me what is problem here. Like, see, in this 50 drugs tested on human volunteers. So in phase 1, failure is 55%. So first we have to find out how much drug has passed in phase 1, right? So 50 drug is there. So 50 minus 50 into 55 upon 100. So 22.5 drug only will pass the phase 1, right? So let's so only this much drug will pass phase 1. So the drug which passed the phase 1, that only will go to phase 2 now. So this much drug we will take in phase 2, and again it's telling the probability of failure in phase 2 is 35.3%. So again 22.5 minus 22.5 into 35.3 upon 100. So it will come now 14.56. Like we have taken 22.5 drug in phase 2, but on that only this much drug will pass, like 14.56 drug. So this much the drug which pass in phase 2, that only will go to the phase 3. So here in phase 3, we will take this much drugs, and again it's telling like the probability of failure in phase 3 is 45%. So 14.56 minus 14.56 into 45 upon 100. So answer will be the 8. The number of drugs we successfully pass all the test is the only 8 drug. Yeah, your liver, kidney, pancreas. Xenobiotic metabolism takes place in, yeah, liver. Yeah, xenobiotics means drug, na, compound which takes from yeah, outside. Just see which of the below is not true. Ah, yeah, both having beta lactam ring, yes. Country lactal infections promote. अरे ये this one promote cell wall synthesis. No, it won't promote cell wall synthesis. No, just now I told show you na like how antibiotic is binding uh, to that region, transpeptidase regions and inhibiting their process, that cell wall synthesis. So this one D. So these all your assignment questions. Okay, okay. What? Which? Because once a drug will pass the phase one, then only it will go to the phase two, no? Yeah, because these all are the different properties. In case, okay, someone has told like these all are the poor pharmaceutical properties, that lack of efficacy and toxicity. If in case of this, like if we will write something else, like some side effects or something, then it will be the independent event, no? Because these all are the different things. But yeah, these all are the different properties of drug, na? Like, how it would be dependent. Like some drugs will show less efficacy, but it won't be toxic. Some drug it is toxic, but it won't like effective. So how it is dependent? All drugs will show the independent, like different properties now. So how it all can say like, and in question it's written like poor biopharmaceutical properties. In this place, we can write some other properties of drug like solubility or something. Then you can understand this question, no? You understand like this? Yeah. That is the confusion. Yeah, that is the confusion. You think like this, like some other properties like solubility or something. Then you can understand, no? But here you have to consider this all as independent event. Or okay, I will discuss these questions again in next class. I will also try to find out. So let's close the class now. Yes. PPT where? Yeah. That NPTEL team will send you. I will send to them and then they will send you. Okay, thank you. So let's close the class now.